Hello students, today our topic is mutagens and their types. Okay, so what are mutagens? Mutagens are the physical or chemical agents that have the ability to induce mutation or increase the frequency of mutation in the DNA. So these are the physical or the chemical agents that have the ability to induce mutation or increase the frequency of the mutation in the DNA. Such agents are called as the mutagens. How do they do this? These agents can directly damage the DNA due to their actions and thereby increasing the frequency of replication errors. These mutations can occur randomly. Various environmental effects play a vital role in their occurrence, and they can be created in lab as well using various chemicals, radiations and physical stimuli. These mutagens are carcinogenic in nature. That means they have the ability to induce cancer in many animal cells, though need not necessarily be in all cases. All mutagens exhibit distinctive mutational signatures, which means they have very specific type of damage caused to the DNA in the type of mutation. So every mutagen has its own distinctive mutational signature or the damage in which it can cause the damage to the DNA. Okay, let's go to the different types of mutagens. Based on origin, mutagens are classified into three distinct forms. They are physical mutagens, chemical mutagens and biological mutagens. Here two main factors play a role in physical mutagens, that is high energy radiations and temperature. High energy radiations can be further classified into ionizing radiations and non-ionizing radiations. We will look into each one of this in detail right now. So let's begin with ionizing radiations. These radiations mainly include X-rays, gamma rays, beta rays, where different elements like electron, protons, neutrons, or any fast moving energy molecule can trigger the radiation, which in turn induces the mutation. Okay, the process by which ionizing radiation can trigger mutation is still mostly unknown. Many changes occur in the chromosomal DNA in the form of deletion, addition of bases, inversion, translocation, or breakage. All these changes can occur in the DNA where at the poly sugar phosphate backbone of the DNA. These changes are induced by these rays, which are due to the electron, proton, or neutron, or any fast moving molecules or the particles, which induces the mutations by either deletion, addition, inversion, translocation, or breakage in the chromosomal DNA. Okay, now let's move to the non-ionizing radiations. Non-ionizing radiations are divided in two types. They are UV rays and visible light. Our main focus as a mutagen is on UV rays. UV rays is a non-ionizing radiation that may cause mutation. The most effective wavelength at which a UV ray can induce mutation in the DNA is 2600 Armstrong. It is at this at this wavelength, the DNA or the proteins maximally observe the UV light or UV rays. Okay. And what are the changes the UV rays causes in the DNA? The changes can be base deletion or strand breakage, cross linkage in the DNA, or formation of nucleotide dimers. Okay, all these are the changes which UV can bring about in the DNA. Okay, now let's look into the different types of UV rays. UV rays types. So based on wavelength, UV is classified into three types, that is UV A rays, UV B rays, and UV C rays. Okay. So here let's see the wavelength. The wavelength of UV rays is 320 nanometers. It is also called as near visible range. Okay, the main effect it causes on the DNA is dimerization of pyrimidines. We know that nucleotides are in two forms, that is purines and pyrimidines. It causes the dimerization of pyrimidines. This in turn prevents formation of replication fork. So when the replication fork is not formed, the replication process of the DNA stops. And why it is stopped? Because of the dimerization occurring due to the UVA rays. Okay, and it has adverse effects on the DNA by stopping its replication. The second type of UV ray is UVB rays, that is having wavelength from 290 to 320 nanometers. This is the most lethal form which can destroy the entire DNA. Okay, this type of UV rays is used in our lab or in our microbial techniques for sterilization. Okay, the third type is UVC rays, having the nanometers, wavelength in nanometers 180 to 290 nanometers. This is mostly filtered by the ozone itself, and it is most lethal, has the carcinogenic effect as well. Okay, so these are the three types of UV rays. Now let's move to the second physical agent, that is temperature. Students, we know that all chemical reactions are influenced by temperature. The rate at which these reaction occurs are dependent on the temperature. Therefore, any increase in 10 degrees of temperature is directly proportional, or it doubles or triples the rate of mutation. That means a 10 degrees rise in temperature can double or triple the rate at which the mutation is occurring. So this is how the effect of temperature is on the rate of mutation. It is directly proportional. It can directly increase the rate of mutation. How does it do? Temperature affects the hydrogen bonds. First, it affects the hydrogen bonds that are in the DNA. Second, it affects the phosphodiester bonds in the DNA. When these two bonds in the DNA are affected, it disturbs or disrupts the stability in the DNA, and thereby contributing to the mutagenesis. Okay. Now let's look into the chemical agents or the chemical mutagens. Okay. So chemical mutagens. These chemical mutagens have effect on the chromosomal DNA either by direct gene change or by copy error. Okay, this is done in the DNA, okay, by the chemical mutagens. So based on these, they are classified into 4 types. First is base analogues, second type is intercalating agents, third is metal ions, and the fourth one is alkylating agents. So these are the 4 types of mutagens that bring about the changes in DNA either through direct gene change or by causing copy error change in the DNA, chromosomal DNA. So now let's look into each one of these types, okay. So base analogues. Now what do you mean by base analogues? Base analogues are those substances that are structurally similar to the bases, like purines or pyrimidines. What are these purine pyrimidines? They are nothing but the nitrogenous bases. We know that, right? Now, what changes do these base analogues bringing? The base analogues actually participates in the DNA replication process. They take up the place of their analogues, okay. What are the changes they bring in this are, they participate in replication process in the place of original base and thereby bringing changes in the DNA. Now let's see this by example, that is 5-bromo-uracil. This is an base analog of thymine, okay. And the another example is aminopurine, which is a base analog of adenine. So whenever the thymine has to participate in replication process, it is this 5-bromo-uracil goes in its place and bonds with the other nucleotide, okay. Similarly, aminopurine in place of adenine, okay, it goes and binds to the another nucleotide. Suppose A has to go and bind with thymine. Instead of adenine, aminopurine goes and binds to the thymine in the replication process. Similarly, it is with the 5-bromo-uracil as well, okay. And thus they bring in the changes in the replication. Now the second one is intercalating agents. Now what are these intercalating agents? These are the agents which have hydrophobic, heterocyclic ring structures. These structures resembles the base pairing ring structures. Okay, these agents themselves get embedded, they themselves get embedded in the DNA helix, causing interference with transcription, replication, and thus causing the mutation. The mutation here is mostly caused by frame shift mutation. Okay, the best examples we can have here is ethidium bromide, or proflavin. These are the examples of intercalating agents. Okay, now let's move to the third type. These are metal ions. Okay, we know that metal ions are mostly producers of reactive oxygen species, right? So metal ions always produces reactive oxygen species, ROS. Many metal ions like nickel, chromium, cobalt, arsenic, cadmium, iron, etc. Because of this reactive oxygen species, there is DNA hypermethylation occurs. Okay, whenever DNA is hypermethylated, the DNA repair mechanism doesn't work at its full efficiency. Okay, DNA repair mechanism requires the DNA to be in unmethylated state for some period of time. But when the DNA is hypermethylated or in the hypermethylation state, the DNA repair mechanism does not work, and therefore this leads to the DNA damage and obstructs the DNA repair process. Okay, and thus the mutation is occurring. Okay, now the fourth type is alkylating agents. So these are the chemicals, they damage the DNA by inducing alkyl groups. So how do they damage the DNA? These alkylating agents damage the DNA by introducing alkaline groups. When these alkaline groups are induced in the DNA, it increases or boosts ionization. When this happens, base pairing mistakes occurs. It causes breakage or holes or gaps in the DNA strand, thereby having direct mutagenic effect. Okay, So what are the examples of this? The examples are nitrous acid, then formaldehyde, and vinyl chloride, and epoxides, etc. So these are the example of alkylating agents. They induce alkyl groups in the DNA, thus damaging it by boosting ionization, which causes base pairing mistakes and which in turn results in holes or gap in the replicated DNA and thus causing direct mutagenic effect. Okay. Now the final type, that is biological mutagens. So they are basically again of three types: transposons, viruses, and bacteria. Okay. So transposons, they are nothing but the fragments of DNA. So what are transposons? Transposons, these are DNA fragments having the ability to self relocate. They have the ability to self relocate and self multiply. Okay. So now let's look at this transposon itself. These are also called as insertion sequences. I mean these are the smallest transposons. They are the smallest ones. Insertion sequence IS is the smallest transposons having base pairs of 10 to 50 base pair length. Okay, they can self relocate or self multiply. Okay, they usually keep on migrating around the DNA. They are usually referred as the jumping genes. They keep on rotating around the DNA and they are called as the jumping genes. The functionality of this gene is that when they are inserted into the DNA, they disrupt the gene. When they get inserted into the DNA, they disrupt the particular gene. This is their main function. Now let's look at the types of transposons. Basically there are again three types. The first one is replicative transposons. These transposons, they retain original locus and translate its copy. Okay, they retain its original locus and translate its copy. The second one is conservative transposon. Here original transposon translocates itself. The original one is itself participating. Okay, here it is making a copy and then participating. Here the original transposon itself translocates itself. Okay, the third one is retro transposon transpose. Retro transposon transpose. They are translocated via RNA intermediates. They don't directly participate, but they are translocated through RNA intermediates. So there are the three types of transposons. So what are the transposons? They are the jumping genes. When induced in the DNA, they bring about infunctionality of the entire gene. They have the ability to self-relocate themselves and self-multiply themselves. Okay, and these are the three types of transposons. Okay, now the second one is the virus as the mutagenic agent. Insertion of viral DNA into the genome may lead to disruption of genetic function. It causes deletion, insertion, point alterations, or base substitutions in the DNA. Okay, due to this, the cells are prone to error. Okay, the best example here is Ross sarcoma virus. Okay, now the last type is the bacteria. Bacteria as the mutagenic agent. So some bacteria produces reactive oxygen species. Okay, they can cause inflammation, and this inflammation leads to DNA damage or decreased DNA repair mechanism. Okay, the best example here is Helicobacter pylori. Okay, so thus we complete the topic mutagenesis.